Okay, so your quiz on Wednesday. You will you need to study this because this is what your quiz is on Wednesday. So your Wednesday, you're going to have a quiz on the cell cycle and mitosis. Okay, so everything we started talking about last week and everything up until today. Tomorrow we're going to talk about the cell cycle and how it relates to cancer and cancer cells. That will be a quiz that you take after Thanksgiving. So the quiz on Wednesday is only up until this stuff that we talk about today. So only the cell cycle and mitosis. That's it. Okay? So this worksheet. So Thursday and Friday, you should have worked on this worksheet that was on Canvas. Yes? All right. So number one, what is meant by the cell cycle or cell division cycle? Yes, Donovan?
I don't think I have contract.
It's the very beginning.
Oh.
Okay. So what is meant by the cell cycle or cell division cycle? It is the series of events that take place between the formation of the cell. So after a cell has divided and it restarts the cycle, because a cycle goes around in a circle. So when it restarts the cycle, that is the beginning of the cell cycle, and all the way until the moment it replicates. So that is the cell cycle. In what type of cells, prokaryotes or eukaryotes, does the cell cycle occur? Your cell cycle and mitosis occurs in your eukaryotes because your prokaryotes use what? What is your prokaryotes use for cell division? What is it called? It's from your notes. Binary. Fission. Fusion is when you fuse two things together. Fission is when you separate things. So binary fission is what your prokaryotes do. Your cell cycle and your mitosis is what your eukaryotes do. All right. Name the two main phases of the cell cycle. So your two main phases are your interphase and your mitosis phase or your mitotic phase. All right, so your interphase and your mitotic phase, those are the two main phases of your cell cycle. Now, you have phases inside each of these. So interphase is made out of 3 stages and your mitotic phase is made out of 4 stages. But these are your two main ones. So either your cell is in interphase or it's in your mitotic phase. Okay. Interphase is between the times when it's dividing. So again, it's either in interphase or it's in your mitotic phase. One or the other. What is occurring in a cell during interphase? So during interphase, you have your G1 phase, your S phase, and your G2 phase. During these phases, your cell is hanging out, being a cell, growing, doing whatever job it's doing. During your S phase, it is going to replicate its DNA. And then during your G2 phase, it's going to get ready for cell division. So you are growing after your previous cell division. You're going to grow, you're going to replicate your DNA, you're going to do some more growing, all the while you are doing whatever job you were told to do and hanging out being a cell. Okay? 2 things that I want you to fix or that I want you to do. Number 5. So during interphase, this is when your DNA is replicating. And then number 6, during mitosis, that is when your cell is dividing. So please fix that on your worksheet for number 5 and 6. Number 5, interphase, that is where your cell is growing, replicating, growing again, and then once your cell has entered the mitotic phase, that is when it's going to divide. So during your mitotic phase, that is when your cell is dividing. So please fix those 2 on your worksheet. And then down here we talked about, or you should have read about, interphase. So interphase. Cells obtain nutrients and duplicate or copy their chromatids or genetic material during interphase. Where are the chromatids found? In the nucleus. And then there's a picture, but I want you to finish getting this. So while you're finished getting that, I'm going to try and just explain this visually to you. So, during your interphase, each cell has, here I'll do one, each cell has one arm of DNA. So this is a cell, this is the DNA that's inside that cell, right? So your interphase, you have your G1, so it's going to grow. Your S phase is when your cell is going to duplicate, and you have 2 copies of your DNA now. It's going to go through your G2 phase, grow a little bit more, and then it's going to go into your mitosis phase or your mitotic phase. During your mitosis phase, your cell's DNA is going to split in half to make 2 new cells. So the key thing that you need to understand about mitosis, if you do not listen to anything else that I say today, please listen now. The key thing about mitosis is that you are going to make 2 identical daughter cells from an original parent cell. So you have 1 strand of DNA. During your S phase inside of your interphase, you're going to replicate that DNA. Then you go through your G2 phase, and then you enter your mitotic phase. Your mitotic phase is going to make 2 new cells. Each of those cells are identical, have the same amount of genetic information as your parent cell. Because your parent cell started with 1 arm of DNA, your 2 daughter cells are going to end with 1 arm of DNA. That is the main thing about mitosis that you need to understand. Okay. So then we had down here, we had a picture of your chromosome. So on your quiz on Wednesday, you will need to know the parts of a chromosome. So this whole thing is a chromosome. Your X is your chromosome. This is after your cell has replicated its DNA. So you had 1 arm. After replication, you had 2 arms. So this is your 2 arms. Okay? So this is your chromosome. You have 4 legs. Each leg is called a chromatid. In your notes, we learned about sister chromatids. Sister chromatids are these 2 legs. So this is 1 pair of sister chromatids. And then you have these 2 legs. This is another pair of sister chromatids. All 4 of your chromatids, or both pairs of your sister chromatids, are conjoined in the middle or at the center, and that center is called the centromere. Centromere, center. So, you have 4 legs. Each leg is called a chromatid. 2 of them paired together is called a sister chromatid. They are all joined at the middle or the center. That is called your centromere. So please know your stuff about your chromosome. Labeling chromosome on your quiz on Wednesday. And then on this side, it had another picture that you had to label the chromosome. But now what we need to realize is, so we have this sister chromatid and this sister chromatid. But what you have is one is longer than the other. So number 1 is pointing to one of your legs. One of your legs is known as your chromatid. All 4 of your chromatids are connected in the middle at your centromere. And then you have 1 pair of sister chromatids. The ones that are shorter are called your short arm. The ones that are longer are called your long arm. So these are your short sister chromatids. These are your long sister chromatids. So again, make sure you know the parts of your chromosome for your quiz on Wednesday. Okay. And then we had some more reading about interphase, and then we had some questions. So in what phase do most cells spend the majority of their lifetime? That is interphase. Remember from your notes, about 90% of your cell's life cycle is spent inside interphase. Okay. How often do human skin cells divide each day? Once, so it is more often than I thought. I thought it was every 2 days, but it is every day. Your skin cells are dividing and replacing each other every day. How many hours per day is a human skin cell in interphase? About 22. What type of cell may spend decades in interphase instead of dividing? Your nerve cells. Name 3 stages in interphase. So interphase is made out of 3 phases. That is G1, S, and G2. For your quiz on Wednesday, I'm not going to ask you specific questions about interphase. I'm not going to ask you, how much time does a cell spend in interphase? I'm not going to ask you that. I am going to ask you, or I could ask you, what phase do your cells spend most of their time in? That is your interphase. So you do need to know that your interphase is a big chunk of your cell's life. And then you do need to know the 3 stages of interphase. So G1, S, and G2. That is interphase. All right. So what is happening during each of those stages? So G1, G stands for growth. So G1 is the next step after your previous cell division. So your cell has just divided, and it is a cycle, so it's going in a circle. So after it finishes dividing, it's going to restart another round of your cell cycle. So it's going to go back into your G1 phase. So your G1 phase is right after the cell has divided. So your G1 phase, it needs to make all the things that your new cell doesn't have. Because once it divides, a lot of your organelles are either going to go to one or the other. So now whatever organelle it's missing, it has to make that organelle. Okay? So it's going to make organelles, it's going to do some growing, it's going to become an actual cell. And then your S phase. S stands for synthesis. So G stood for growth, S stands for synthesis, and you need to know that synthesis means to make. So synthesis, you are going to make a copy of your DNA. So your synthesis is when you are going to copy your DNA. And then your G2 phase, G stands for growth. So you're going to grow a little bit more, but now you are growing in order to get ready for mitosis. So this part, this G stage, this G1 stage, is growing because you just divided. So you need to grow a little bit. You need to make the organelles that you're missing. You need to get ready to be an actual cell. This G2 phase, okay, now you are an actual cell, but you have just replicated your DNA, which means you are ready to make new cells. So you need to grow a little bit more to get ready for another round of cell division. So growing, DNA synthesizing or copying DNA, growing again, and then you enter your mitosis phases. Again, I will have this key on Canvas tonight, so I'm trying to get through this pretty quickly so that we can move on tomorrow. If you miss anything, the key will be on Canvas tonight so that you can get those answers tonight, okay? Real quick, side note, if you ever need to get anything off Canvas, please go under modules, because that is how I organize everything. If I tell you that everything is on Canvas, then when you get into the course, go straight to modules, find the unit, and then everything is in order for you. So if you're wondering what order things are going to go in, if you need to turn in your notebook from last unit, but you're not sure what order the things go into the Canvas course, go under modules, look under unit 3, and it is numbered 1 through, I think there was 12 things. So 1 through 12, and it will tell you what each number is. Click on it, and it will tell you, and it will have links, it will have YouTube videos, whatever you need for that assignment or for that set of notes, whatever it is. But everything is under modules if you ever need to go into Canvas to get anything. All right. So these questions, we will talk about this starting tomorrow when we talk about cancer, because this is actually kind of what happens when you have cancer cells. So real quick, what stage of the cell cycle do cells sometimes exit? They exit the G1 stage. And if they do exit the G1 stage, they go into what's called the G0 stage. That is where you have a G and then a 0 afterwards. If you ever have a 0 after a letter and it's a small 0, it's whatever the letter is and then not. So this is our G0 stage. You could also call it your G0 stage, but it is called your G0 stage. Your G0 stage is a kind of weird limbo land where your cell is still alive and it's still doing its job, but it's not dividing. So it's kind of this weird in between stage. And we'll talk about that starting tomorrow, what exactly that means. So 3 types of cells that enter the G0 phase when they are mature: your heart muscle cells, your eye cells, and your brain cells. And then what happens if these cells are damaged during your lifetime? They cannot be replaced. So your nerve cells, I thought it said nerve cells. But anyway, your nerve cells, if you have nerve damage, then your nerve cells cannot replace themselves. So once you damage those cells, that's it. I thought it said nerve cells. But anyway, we'll talk about more of this when we tomorrow when we start talking about cancer. Okay. And then we started talking about the individual stages of your interphase. So G1. So G1 is your first step after cytokinesis. So cytokinesis is your last step, and then the cycle restarts itself into the G1 phase. So during cytokinesis, the previous step, that is when your cytoplasm divides. So cytokinesis, cytoplasm, cyto and cyto. So cytokinesis is when your cytoplasm divides. When you make new cells, those new cells are now called your daughter cells. So you had the parent cell making 2 new daughter cells, and those 2 new daughter cells are identical. So you had 1 arm of DNA before, during interphase, you replicated your arm of DNA, you made your X, and then your cell divided, and now you have an arm of DNA per cell. So you started with an arm of DNA, you end with an arm of DNA. So mitosis creates identical daughter cells. What is the major thing happening to a cell during G1? Growth. Your G stands for growth. What cell structures are being made during G1? Organelles. Since proteins and enzymes are being made during G1, there's a great amount of protein synthesis occurring. So your cell is growing and making things that it needs in order to be a cell, because it's missing some organelles. Oh, here, there. Okay, so then you have your S phase. So your S phase stands for your synthesis phase, and what you are synthesizing, or what you are making, is a new copy of your DNA. So you had 1 arm of DNA, you undergo synthesis, and that is when you make your X, and you have 2 copies. That is when you make your chromosomes. Right? What happens during the S phase? DNA synthesis or replication. So you're making a copy of your DNA. Each chromosome originally is made out of how many DNA molecules, and how does this molecule appear in the chromosome? So again, you have 1 cell has 1 strand of DNA. So you have 1 strand of DNA. After synthesis, you make 2 strands of DNA. Those strands of DNA, when they are ready to divide, they are going to coil. They're going to condense, smoosh together. So they're going to coil. So they're going to be little squigglies. And they're going to condense and kind of smoosh together to make tightly packed chromosomes. Okay. At the end of S phase, each chromosome has how many coiled DNA molecules? 2. So you started with 1, you underwent synthesis, and you made 2, and those 2 are going to form an X, and that is where your chromosome is made. Yes? We all understand that? Perfect. Okay. Next. So up here, what structure holds the duplicated chromosomes together and is also copied during the S phase? This is called, do not use the word centromere or centrosome. I want you to use the word centromere. Centromere. Centrosome and centromere are the same thing. So you still have the prefix center, which means the center of your chromosome. So centrosome means the center of your chromosome. but centromere is the exact same thing. But I am going to use the word centromere. But you could see the word centrosome on the EOC. So if you do see the word centrosome, it is the same thing as the centromere. It is the center of your chromosome. Okay? And then we talked about, or then you had some reading about the G2 phase. So the G2 phase is the final and shortest phase of interphase. It lasts between 4 and 5 hours. Again, you will not need to know the specific time length of your phases. But during your G2 phase, again, G stands for growth. So you're going to do a little bit more growing. But now, instead of making organelles, because now we have all of our organelles, now we are going to make enzymes that are needed for the cell division. What follows the G2 phase? Mitosis. So after you grow, after you synthesize, and after you grow again, that is when you enter your mitotic phase or mitosis. And then, did everyone get this? Everyone change this up here? All right. So after you enter your mitosis phase, what part of the cell is actually dividing in mitosis? So mitosis is this stage here. Mitosis is when your genetic material divides, but your actual cell, so the cytoplasm, doesn't divide until it enters your cytokinesis. So cytokinesis, cytoplasm. So your genetic material is dividing here. Your actual cells, so your cytoplasm of your cells, are dividing here. And then again, you re-enter your cell cycle with your G1 phase. So it goes like this, over and over and over again. So I want you to now take a few minutes to color code this picture. Okay? Color code this picture. So this is outlining. You have a red line outlining all 3 of these phases, because these three phases make up your interphase, and then your mitosis phase, and then your cytokinesis phase. So take a few minutes now. Come get some coloring pencils if you need them. But you need to know the cell cycle. So you need to color code this and outline your interphase, and know your mitosis phase, and know your cytokinesis phase. So take a few minutes now to color code that picture. If you do not have this worksheet and you are putting your answers on another sheet of paper, please draw this cell cycle. You do need to know the cell cycle. So your quiz on Wednesday is on the cell cycle and mitosis. Quiz on Wednesday, cell cycle and mitosis. So please study this packet tonight and tomorrow night so that you will be ready for your quiz on Wednesday on cell cycle and mitosis. So, give you like 1 more minute, but we need to move on because I do want to finish this before you leave today. So if you do not finish color coding and labeling this cell cycle, please do that tonight. I will have the key on Canvas for you. we are going to move on. Again, the key will be on canvas for you. So let's finish this up. So the last page of mitosis talks about your, actual part of mitosis. Okay, so when are chromosomes duplicated? Before or during mitosis? They are duplicated before, during which phase? Which phase is your chromosomes duplicated? Synthesis phase, your S phase, right? So that is before mitosis, during your S phase. What process follows mitosis? That is cytokinesis. So mitosis is where your nucleus is going to divide, so your genetic material divides. And then cytokinesis is after that. Cytokinesis is when your cytoplasm divides. So cytokinesis, cytoplasm, cyto. How do the two new cells compare with each other? So after mitosis, after a cycle of mitosis, you will form two new daughter cells that are identical to the parent cell. So you are making identical daughter cells each time you do mitosis. The same genetic material, the same amount of genetic material. So two new daughter cells that are identical to the parent cell. Does mitosis occur in prokaryotes? No, it does not, because prokaryotes do not have a nucleus. So they do not need to undergo mitosis. They just copy and paste their DNA. They make a new bacteria cell and they go on their way. And they undergo binary fission. So prokaryotes use binary fission. Eukaryotes use mitosis in the cell cycle. Your body cells are called somatic cells. And your somatic cells are going to use mitosis. They are going to replicate and make exact copies of their cells. Your reproductive cells are known as germ cells, and they're going to undergo what's called meiosis or meiosis. And we will talk about that when you come back from Thanksgiving. Okay? But somatic cells undergo mitosis. They make the same cell over and over and over again. Germ cells undergo meiosis, and they make half of the original cell. So we'll talk about that when you get back from Thanksgiving. Okay? And then inside of your mitosis, your mitotic phase, you have 4 phases inside of mitosis. They are prophase, metaphase, anaphase, and telophase. And then after your 4 phases of mitosis, you have cytokinesis. And your cytokinesis differs if you have a plant cell versus an animal cell. In an animal cell, you have what's called a cleavage furrow. So your cell membrane is going to go in on itself and pinch itself off and then divide to make 2 new cells. Your plant cells are going to make what's called a cell plate. So what it's going to do is after your nucleus of your plant cell has split and divided, your cell is going to form a temporary barrier between the 2 cells called your cell plate. And then that serves as a template to where they form the cell wall on later. So your animal cells create a cleavage furrow. Your plant cells make a cell plate. Okay. And then this page. So genetic material is called chromatin during the interphase, and it is not clearly visible. So during your interphase, you have long strands of chromatin. And then after it replicates, and then while it's ready to be divided, it's going to coil up, and shrink, and get more tightly, densely packed, and that's when it makes your chromosomes. So what makes chromosomes become more visible? They are shortened, condensed, and coiled. What is used to help break down the nuclear membrane? Enzymes. Besides the nucleus, what else is broken down? Your nucleolus. Your prophase is going to make what are called spindle fibers. So your spindle fibers are going to grow out and attach them to the chromosomes and then pull the chromosomes apart. Your centrosomes or your chromosomes are held together inside the centromere. Your chromosomes line up in the middle, so metaphase starts with M, middle starts with M. So the middle of your cell, that's where your metaphase is. Your last two parts are your anaphase. That's when your chromosomes split. And then telophase is where they move to opposite ends of the cells. And then these four things are happening during the last stage. So basically, your cell is going back to normal. And then the very end, you had a lot of coloring and labeling to do. So your last page should look like this. I will have it on canvas for you, but this is what your last page should look like. So please make sure your last page looks like this. Bye guys, have a good day.